Hello everyone, um, welcome to today's webinar brought to you by nQuery from Dotmatics. Today we're going to be looking back at some of this year's most popular webinars on clinical trial design topics. I'm going to be giving a quick run through of 3 different webinars from the past year. So my name is Calvin O'Brien and I'm a research statistician here working on nQuery, and I'll be delivering today's webinar. So the topics we're going to be revisit today are survival analysis designs, complex group sequential designs, and non-parametric tests. These were 3 of the most popular webinars from the past year. So the plan is to give a quick overview of what each of them entailed, along with exploring some of the examples provided in each webinar. If anyone is interested more in any of the topics, there are full webinars of each available on the nQuery website. Especially with the group sequential designs, there's a lot of different webinars available, ranging from introductions to what we're going to be discussing today, which is more complex group sequential designs. So if you're unfamiliar with any of the topics, I'll be giving an introduction to each, so there isn't much of a prerequisite knowledge on any of the topics required. And again, yeah, if you're interested in a more thorough introduction, I would definitely recommend checking out each of the webinars. These were performed by my colleagues Paul, Brian, and Dennis, so there's a wide range of expertise brought into each of the sessions. As always, questions are welcome throughout the webinar, and if there's any issues, you can let us know using the chat function. After the webinar, we'll be emailing out the slide deck, so if you're interested in any of the resources within, you'll have access to that after, or they can also be downloaded from the website a few days after. They'll be under the web player. As I mentioned, this webinar is brought to you by nQuery from Dotmatics, the complete solution for optimizing your clinical trial designs. So the examples today are going to be worked through nQuery. nQuery is widely used in the pharmaceuticals and biotechnology industries for power and sample size calculations, and has been on the market for well over 25 years. nQuery is known to make clinical trials faster, less costly, and more successful. As you can see from the infographic, there are methods available across all the phases of the clinical trial process, but the main focus here being on phase 3, confirmatory phase 3 trial designs. So nQuery operates on a tiered licensing model. So today we're going to be demonstrating some examples across a few different tiers. If you're interested in viewing what's available for each tier, you can use the package comparison tool, which is linked at the bottom of the slide, or you can go onto the nQuery website as well to find that. This is a progressive model, so each tier has the features available to all the lower tiers, plus whatever that tier itself brings to the table. All customers have access to the basic tier, which focuses on sample size and power calculations for fixed term trials, plus then focuses on stuff like credible intervals and topics like Bayesian assurance. Pro then is our most popular tier, I would say. This includes a lot of the adaptive designs, like group sequential design. So a lot of those features we'll see today are from that. And there's other adaptive stuff like MAMS models, the multi-arm, multi-stage models, and stuff like that. Finally then, we have our expert tier, which currently, the focus is on milestone prediction for enrollment and survival event targets. Just to note, I won't be discussing any of that today when it comes to the survival analysis. This is more introduction that's a bit advanced for that. So if you're new to nQuery, or if you have a subscription but you want to check out some of the features in the higher tiers, you can get a free trial of the full software from the nQuery website. And there'll be a link available at the end of the slide deck as well for anyone interested. So with that, that's the end of the housekeeping. And we'll jump into the first of today's webinar recaps, which is on survival analysis designs. This webinar was delivered by my colleague Paul back in May of this year, where he goes into detail about what survival analysis is, some of the decisions and issues that can arise when designing survival trials, and how to handle scenarios where certain assumptions are not met. So that focus mainly today will be on non-proportional hazards. So I'll jump straight into it now and just give a brief overview of what was discussed. So survival analysis, or also known as time to event analysis, is performed when you want to make inferences about the expected length of time until an event of interest occurs. So your interest here is then the length of time it takes for a subject to experience an event. We can define an event depending on our needs. So if we're interested in survivability, death would be our event, and our endpoint would be overall survival, or OS. This is considered the gold standard when it comes to survival analysis. You'll see this reported a lot. But we can also use other events, such as for disease progression. So in that case, we'll use progression-free survival, or PFS. These are two of the primary endpoints you would see in survival analysis trials. But you're not just restricted to these. Again, it's however you define your events. So if you're interested in alleviating symptoms for a chronic illness, you might look at something like symptom expression. You might also see stuff like this if you were doing like a multiple endpoint trial, or doing something like gatekeeping, where you would test your first set of hypotheses first, like death or overall survivability and progression-free survival. And then afterwards, you would check stuff like symptom expression in your second set of hypotheses. So given these endpoints and kind of how survival analysis is framed, it's clear to see why this would be a highly useful tool, or a highly useful way of performing analysis in a field like oncology, where you would often have different stages of cancer, and you're interested in preventing progression onto the next stage of cancer for as long as possible. Or if you're dealing with a more aggressive form of cancer, your interest might be in focusing on or just extending overall survivability. In early stages of oncology trials, you might see them use response rates rather than going through all the rigmarole of performing survival analysis. So this is more of a in line with proportions rather than time to event. The reason being is due to a lot of the uncertainty that surrounds survival analysis, and also because a lot of these trials, well, that ties in, that a lot of these trials at early stages are probably going to stop due to futility. So it's not until maybe your phase 3 trial, or phase 2 or phase 3 stage, that you're interested in going all out and performing your survival analysis. So when designing a survival analysis trial, there's a lot of different decisions that need to be made, which will drive then how much power you have available. And this is essentially true for any clinical trial you're running, but especially for survival analysis due to all the different uncertainty that'll surround it. So here we see 4 different decisions that you might need to make. Or not it might, but these are 4 primary focuses you're probably going to be looking at. So design is one. Typically, you'd probably think of your parallel trial design, your case cohort, where you've got your control group and your treatment group, and you want to compare results from the 2 groups. You might also have one-arm trials, whether that be could be ethical or logistical or financial reasons, that you only have one group to compare. And lastly, then you might then have more complex designs, which we'll touch on later. So the likes of group sequential design, or you could even have multi-arm, multi-stage trials where you drop off underperforming arms and keep strong arms when testing different treatments. Another decision you might have to make then is what statistical method you're going to use. So here we've shown log rank and Cox. Traditionally, these were probably 2 of the most primary methods used when it came to survival analysis, but now we're starting to see a lot more flexible methods come on the scene. Paul does a good job of going through these in his webinar. So you'll have stuff like weighted tests, which put more emphasis on cases at different stages of the trial, or another max combo would be another one, which I'll discuss, and Paul does a good job of discussing as well in his webinar. So the decision you make in your statistical method is most likely going to dictate your endpoint, how you're going to be reporting your results. So here we see hazard ratio and survival time. Survival time is fairly straightforward, just how long the expected survivability is. Hazard ratio then is the ratio of the instantaneous event rate, or the hazard, I suppose, between groups of an event occurring. And this is what a lot of survival analysis trials report. Lastly, then, is how you want to describe your survival distribution. So the simplest way of doing this is defining it using the exponential distribution, but there's other distributions available, like listed here, the Weibull distribution. And this allows you, if you were interested in adding a time varying hazard rate. And this ties a lot into calculating your sample size from events, which I'll touch on now in a second. So each of these are going to affect your power or sample size calculations. So yeah, like I said, it's important to note that unlike your usual, say your two-mean test or whatnot, that your power is going to be, or your information, let's say, is driven from your sample size. So again, bigger sample size, more power and whatnot. But in this case, sample size isn't the direct feeder of information. We're more interested in, well, we are just interested in events here. Of course, events come from sample size, but our calculations would be for number of events. And the tricky part then in dealing with survival analysis is when dealing with events, we know how many events we're going to want to see, but we're not guaranteed to see, you know, however many events from a certain amount of subjects in a specific time. We can go off and calculate using all our data and like our survival distribution and whatnot, kind of the expected amount of events at a given time from a certain sample. But again, there's no way of truly knowing how long that's going to take. So there's a good level of uncertainty that surrounds this. This is further complicated then when dealing with stuff like accrual and dropout. If you've got kind of a non-constant accrual rate, or you've got different dropouts between the groups and whatnot, this can further complicate your sample size or how many events you're going to have at the end of the trial. And then this is especially complicated then when you start introducing stuff like group sequential design, hence why it's listed under here as the complex trial design. So I'm going to jump into our first example now, and this is an example Paul goes through in his webinar. We're going to stick with this for the next few examples, just because it's a handy way to compare the different methods and as we go on. So in this trial, they're performing a log rank test. There's a one-sided 2.5% significance level, and we can see here we need 282 events rather than sample size to allow for 92.6% power to show a risk ratio of 2/3, and that's calculated from our median performance or progression-free survival of 6 months and 9 months in the control and treatment group respectively. And then we see information here about uniform accrual, so that's nice rather than complicating that. So we see over 74 weeks, that's our accrual period, and we do have a minimum follow-up of 39 weeks. And we assume an exponential survival distribution, and so from that we can calculate the amount of patients we'd need, and we see in this case we need 352, and then they also account for a small bit of dropout. So they add that, or they account for 10% loss. So we're going to swap out or hop out of this for a second, if I can full screen and go back a slide. And we're going to swap over to nQuery now and just go through this example. So when you open up nQuery, brought to this screen, when dealing with most of your power sample size calculations, you're going to be coming here to create a new table, or you also have that option up here. And we'll come here then, and we will look for our table. So we're performing a 2-sample log rank test. And we mentioned we're looking for exponential survival. And you can see there's different tables available for different assumptions. So if you're assuming for piecewise accrual rates, hazard rates, and dropout rates. So as I mentioned, if you've got more complex information on that, tables like this would be more appropriate. But for now, we're keeping it simple. We're just going to do regular, we're not assuming any piecewise information. So when you open up a table in nQuery, you'll be brought to the screen, and you'll see this like spreadsheet-like interface, which is, I would say, fairly straightforward. Each column being a different calculation, so it makes it nice to compare different calculations, whether you're performing stuff like sensitivity analysis and whatnot. So we'll start inputting our information, and when we put in, there's a nice little helper thing here on the right, just explaining to us if we're confused on anything. So 2.5% significance on a one-sided test. Like Paul does in his webinar, I'm going to convert the information here from weeks to months, just to keep in time with our hazard ratio information of 6 months, 9 months. So I think we had an accrual period of 74 weeks, which I think translated to 18.5 months. That's our accrual period. And then our maximum length of follow-up is going to be the length of our accrual period plus the minimum length of follow-up. And we can think of that, we can see it described here being, we assume if someone's recruited at the start, they're going to be measured for this entire time. And if someone was recruited at the very end of the accrual period, they're going to be followed up just for the minimum follow-up time. So that was 39 weeks, which I think comes out to 9.25 months. So adding the two of them together gives us 28.25. And so when you come down to these, we could easily just enter our hazard ratio, or if we didn't have that information, we can use this down here, which is just that side table here in nQuery, and we can go in here and we can type in our information. So if I type in 9, or we got put in 6 here for median survival, we put in 9 for this one, or yeah, I knew I was doing something there. 9 and 6. If I got it right, that would help. There we go. So yeah, we can see our hazard ratio then calculated from this as 0.667. The other way around, we would just have the hazard ratio 1.5, and it being just the same test except in the opposite direction. Just we would just be assuming that our treatment has a negative effect. So once we've all this calculated, we can transfer this up to the table, and we can see it just pops it in up there. And now if we say, well, we said what in the test that we wanted a 92.6% power, put this in, and we get back our figure here of 176, and that multiplied by 2, it gives us our 352 that we saw in the example. And we can also see here nicely that we can also see displayed the number of events required. So again, this is coming from the fact we're assuming an exponential survival curve. So given this would actually calculate this total number of events required, and then from the exponential survival curve and some of the information here, that's where we're actually getting our sample size per group. So I'm just going to go back to this now. Just do full screen. So that's that example covered, and we can see how we can recreate the results. So now I will discuss something that was briefly mentioned at the start, which is non-proportional hazards. So there's a lot of, as mentioned, there's a lot of different decision making when it comes to methods you want to use. I mentioned log rank and Cox, but you can have issues that arise with these when you have non-proportional hazards. So log rank and Cox assume that the gap between survival rates remains proportional throughout the trial, or that the hazard ratio remains constant throughout the trial. But you can see on the right 3 different scenarios where this isn't the case, where you have a delayed treatment effect. You would see maybe a diminishing treatment effect where the treatment wears off over time, or you have crossing hazard as well. So crossing hazard you can see here as having a negative effect at the start on survivability, but as time goes in the long run, it has a positive effect on survivability. So in all of these cases, you have non-proportional hazards. We're seeing these complex patterns more often in practice for a lot of different reasons. For example, many immunotherapies in oncology have have been shown to have a delayed effect, where the drug doesn't take effect until a certain period of time has elapsed. We have that due to issues as relying on the immune system to ramp up, or to the greater effect of non-responders. This is in comparison to the quick effect of traditional cytotoxic treatments such as chemotherapy. For scenarios like these, our power is going to be a lot lower, it's just going to be lower than reported due to not meeting the assumptions of the statistical methods. And there's a lot of discussion now on how to handle non-proportional hazards. So in an ideal world, you would have one method really that would be robust enough to handle all these different scenarios. So here is just a few different methods that can be used when it comes to non-proportional hazards. You can see here at the top, you just use your log rank, where you take your average hazard ratio. But again, it's been discussed that you're not fully meeting the assumptions of this trial. Like, it's not well suited to this. So here are a list of other methods that could be used. I mentioned earlier about weighted tests and whatnot. I won't go into detail about these because Paul does a nice job at it in his webinar, and just for time's sake, just to save time. But I will focus on one of them here, which I mentioned earlier, as being max combo. So max combo is a method to compare multiple tests at the same time while accounting for type 1 error inflation. So when using the max combo, the Fleming-Harrington method is the most common used, as it's equivalent to multiple different trials depending on its parameters. So when both values are set to 0, you have the log rank test. When you set the parameters to 1 and 0, you have the peto test. And listed here on the right is just a few different literatures, stuff in existing literature on recommendations for what tests you might want to compare. And just to note, there will be a list of references available at the very, very end of the slide deck for anyone who's looking for resources. So you can see here that typically you'll see 2 to 4 candidate tests considered, and different recommendations here. Max combo isn't perfect, however. There are criticism of how well it controls the type 1 error rate. There are some cases where it'll falsely report on significance, even when the treatment is always worse or equal. McGurr's modestly weighted linear rank test is one proposal which may alleviate these issues for the delayed effect scenario. And this was added as an option into nQuery's max combo and linear rank tables in our most recent update, the 9.4 summer update. There's also issues then around interpretability and how you report your piecewise hazard ratio using this method. One suggestion is to use max combo for your analysis, but then when it comes to reporting, use something like the restricted mean survival time. So we're going to revisit our first example, except this time we're going to change the assumptions a little. This time we're going to assume that for the first 6 months of the study that there is no effect, that we have a delayed treatment effect, that for the first 6 months our hazard ratio is just 1, and then after those 6 months our regular study will resume that our hazard ratio of 0 or 2/3 will kick in. So I'm just going to swap back here to nQuery, and if I look up here, max combo, here we go. You can see the 3 different tables just depending on what your hypothesis is. In our case, we're just going to use this one here. So yeah, we can see this fits what we're looking to use, our piecewise survival and maximum combination linear rank tests. So we're going to re-enter our details and similar enough to our first one, so 0.025, a one-sided test. We're going to perform 4 tests out of interest. So we can see the 4 we're going to perform over here in the number test. We can also see it here down at the bottom of the side table, and we could change these parameters if we wanted. But we're happy enough with them anyways. They're a nice set based off some existing literature. There's recommendations to use this, so we're just going to stick with it for the time being. So number of periods, we're going to say 3. say our 6 month, our 18 month, and our end of trial. So our initial accrual period, initial accrual portion, that's just 0. We're assuming there's no one in the trial before it begins. Accrual period then, regular 18.5, and we're still assuming uniform accrual, so we're going to set this to 0. Maximum follow-up period 28.25, carrying that over from the assets, and sample size ratio of 1. So this brings up our piecewise side table now for entering our hazard rates to different stages. So we're into 6 and 8.5. Again, I have awful difficulty with these cells. So we're going to assume 1 here, and then we're going to assume our 2/3 here. Now let's make sure I get this right, because I do have a small bit of difficulty with getting these in. So we'll see now. So we're going to put this lower. Yeah, that seems to be right. The look of the draw. So yeah, as we can see, our information is just auto-calculated itself. So we can see our hazard rate for our control group here. I actually just need to change this now because I can't get everything right. And we'll just take this. It'd be silly to assume that. Of course. There we go. I jinxed it now, so that's on me. Clear these and type these in for myself. No, that's a bit better. That tidies it up a bit, I hope. Yeah, that all looks right. And then we mentioned we're not assuming any dropout in this case, so zeros. And we're going to use our sample size from this. So 176, but we're assuming the total sample size, so 352. Make sure that figure is right, 176, 352. And we can see our power calculation then comes out to be 38.81. So in doing this scenario where there's a delayed treatment effect, we can see a significant drop in power, or I suppose inversely, we can see, or related, we can see a requirement for a much larger sample size compared to the case where we just have constant proportional, or we would just have proportional hazards. We can actually come up here to our number of tests, and we can see the individual powers for each of the tests. So we can see our Fleming-Harrington when the parameters are 0 and 1 being our strongest, here being 48.542, and our 1, 0 being quite a weak test in this scenario. But the advantage of doing a test like max combo is that you are running a much more flexible test, that if your if your piecewise hazard ratio didn't follow exactly as to what you thought and was somewhat different, that you would not just be stuck using this one test. And we can actually see this here. I have this done in this table here, where this is how our sample size to get a 90% power using just that strongest Fleming-Harrington test. So we can see we'd need just over 1,500 per group in order to get 90%. And by using our log rank, we would need just shy of 2,000 total to get that 90% power. So again, if you were dead set certain that your hazard ratio was going to be distributed like that, it's going to follow that pattern, you might like to just stick with this and have the lower sample size. But if we come back to this table here, and if I just swap this to the sample size calculator, and I suppose, yeah, I should update our power to 90, that would help. So we can see just shy of 600 per group is what we would need to get 90%. And when we compare that back to our linear rank test, we can see we're basically just requiring a small bit under 100 extra patients per group in order to add this flexibility to account for potentially other piecewise hazard ratio distributions. So it's not well, depending obviously on circumstance, but it's not a massive big jump in sample size in order to add that flexibility. And we can see here when we come to our different tests, we can see our Fleming-Harrington with these parameters is performing quite strong. And we can really see now the weakness of this Fleming-Harrington test with this piecewise hazard ratio distribution. So that's all I really want to say on Max Combo, just that it's a nice tool to use that adds a lot of flexibility into your testing rather than just being restricted if you have uncertainty around your hazard ratio, how that's going to take shape. I'll swap back now to the slideshow. So that's all I really want to say on just the intro to survival analysis. We're going to move now, not away from survival analysis, but we're going to now discuss Brian's webinar, which was done in June, which was complex group sequential design. And this is going to discuss our complex survival analysis mainly. So to give an introduction to group sequential design as a whole, there are a subset of sequential designs. You can have either fully sequential designs where the trial is evaluated continuously as data comes in, or you can have group sequential designs, which where you perform interim analysis at pre-specified points, such as after a length of time or when a certain amount of information has come in. The biggest appeal to these types of trials is their ability to stop for efficacy or futility. So if your trial is performing particularly well at any of the interim analysis, you can stop for efficacy rather than needing to see the trial out. And the same can be done in the alternative scenario where your trial is performing quite poorly, and rather than wasting resources seeing the trial out, that you can stop it for futility. And this is what was done for a lot of the COVID trials, that they were done using some group sequential design, and a lot of them then were stopped for efficacy early rather than having to see out their entire trial. So this allows for a trial to have multiple chances of succeeding. And so if you do this naively, your type 1 error will be inflated. So for example, there's a seminal Armitage paper that shows that a standard group sequential design with 5 looks can inflate the chance of a type 1 error occurring from 5% to 14%. So that's almost tripling the likelihood of making a type 1 error. So it's really important to account for this error inflation. And luckily, there's a lot of different ways of doing this with group sequential design. In practice, group sequential designs are far more common in actual clinical trials than fully sequential designs due to the logistical burden on continuous monitoring. So for example, the greater involvement of the Independent Data Monitoring Committee, IDMC, or the increased chance of operational bias due to unblinding. It can also be shown that for most scenarios, a handful of interim analysis captures most of the efficiency gain, so i.e. the lower average sample sizes of a sequential design compared to one with a large number of looks. So this slide has been included here just to show it off, but it's been discussed in multiple other webinars. So for time's sake, I'll just briefly explain the idea behind error spending. So this first method here is a way of how you would do a group sequential design. So error spending uses an approach where some of the error is spent at each interim look. So you would use your alpha for efficacy and beta for futility. And this leaves less of the error available then to be allocated for the final analysis. However, so your maximum sample size for the overall trial might be higher, but then you might save said resources if you're able to stop, whether it be for efficacy or futility. So as listed here, there's a lot of other methods available for boundary calculations, but I'm not going to go into the rest of these points as they've been covered in depth already. We'll jump into now some complex group sequential designs. And so we're back at looking at our survival analysis designs. But now we introduced group sequential design to it. So if it hasn't been demonstrated already, there's a lot of complex decisions that go into survival analysis design to begin with. So when you add the GSD framework to it, you only complicate it further. I didn't really discuss it much in the previous webinar, but one of the decisions you need to make when doing your survival analysis is how you want to follow up on your patients. If there's a fixed observation period for patients, as we saw in the last example, where they're monitored for a minimum period, or is it until the end of the study? So these will affect when your interim analysis occurs. As in the group sequential design framework, the interim analysis are dependent on the amount of statistical information available. So you'll be basing it off how many events have you at a certain stage. In the 2means GSD, this information instead comes from sample size, but now we're getting it from cases. So our stages of analysis are defined when we reach a certain number of cases. So that adds a little level of uncertainty as to when these interim analyses are going to occur, because again, all we have our expected time to have this amount of events. Non-proportional hazards, they rear their head again here, where we already showed how to potentially handle them in the simple design. Handling them here in the group sequential design setting is not as well documented as the regular case. Then you have dealing with your accrual, survival, and dropout rates again. So unlike the 2means GSD, it's a lot more difficult to predict exactly when your analysis is going to occur due to just the uncertainty around expected survivability. So when you add complex accrual and dropout rates, you make this process a lot harder to predict. And so it makes it extremely difficult to plan effectively when your analysis are going to be performed. And so this can put a lot of pressure on your trial, whether it be logistically or financially. So as you can see, there's a lot more to be considered when using group sequential design with survival analysis. Just to touch then on the second point here, another type of complex group sequential design are adaptive group sequential design. So anytime you mention adaptive, things kind of get a bit tricky. In these trials, there's some aspects of the trial that you can modify while it's going on. So as mentioned before, you MAMS, the multi-arm, multi-stages, you can have that with group sequential design. It's similar to using the drop the losers approach. Again, with group sequential designs, you're already trying to account for the type 1 error. So adding MAMS and it's how it tries to account for type 1 error, you're combining the two, so you make this quite a complicated process. Other things that might be considered in adaptive group sequential designs, sample size, re-estimations. I discussed this in August's webinar on how to choose your effect size. I discussed this just briefly there at the end. The idea being here that you can perform sample size re-estimations if your estimated effect size is not being achieved in reality, but you still would have a clinically significant result. So you can bulk up your sample size in order to still achieve a statistically significant result. In group sequential designs, you'll typically have unblinded sample size, a re-estimation. So this adds complexity to whether there's going to be a need to recruit new subjects in the study, and then how are you going to handle your operation bias when it comes to unblinding. So it can be quite difficult to plan your study when you don't know when your interim analysis are going to occur. So this may mean that using the group sequential methods, which are more flexible to deviations from the original plan, such as error spending, may be more appropriate for survival sequential trials. Other complications may be the possibility of changing the endpoints of interest between the interim and final analysis. For example, regulatory agencies have suggested that accelerated approval could be made on PFS, your progression-free survival, but final approval would still need to be based on the gold standard overall survival endpoint. And this could be integrated into a sequential trial. Non-proportional hazards, as mentioned earlier, there's still an issue here in our sequential survival trials, as this leads to interim timings no longer being directly proportional to the events. Strategies for dealing with this problem are an active topic in discussion and are related to a wider discussion on best methods for dealing with non-proportional hazards, as discussed in part 1. If you're planning to perform unblinded sample size re-estimation, that also needs to be planned and accounted for. Friedland and Korn point out that events may be increased in two ways. You can either increase the sample size or you can increase the study length. But they point out that the choice between these strategies could be biased, since the treatment seems more effective early in the follow-up. The sponsor may prefer to increase the sample size, since those subjects would be at the start of the follow-up. And conversely, increasing study length might be preferred if the treatment looks better late in the follow-up, since the remaining subjects will mostly already had significant follow-up. So plenty to consider for both standard group sequential survival trials using the log rank test, for example, and some extensions to more complex designs. We're now going to return to that example we've been using for the past while, except now we're going to look at turning this into a group sequential design study and see what effect this has on our results. So these are the changes we're going to make in our example. So looking at 5 equally spaced looks, and we're stopping for efficacy and futility included, and some other details such as our piecewise hazard rates and whatnot. We can see here which we're using, well, what our spending functions are for our alpha and beta. So I'll go over to nQuery now and show off. So this is definitely a big jump up. We're also including different dropout stuff. So to save time, I have these pre-filled. Brian does go through these in his webinar. So if you're looking for the intricacies of filling this out and the whatnot, I would go check that out. But when you fill this out, it's the same as filling out a normal table. You would fill out some details here about your GST parameters. So here we see I've labeled the alpha spending function, the beta spending function, and the parameters for each of these. We can see our information times if we're deciding we wanted to perform analysis, maybe not at the start of the study, but rather in the middle and focus more on the end of it. Or if we didn't want to consider stopping for either efficacy or futility at different stages of the trial. And then just bounds, these would appear after we make our calculation. These would be empty initially. We also see, similar to our max combo table, our piecewise information here. In this case, this is the reason why there's 2 columns here. One of these is accounting for dropout, and the other one isn't. For this table, I haven't mentioned dropout. The only thing significantly different, I suppose, besides the group sequential design aspect, is I've said here that the accrual isn't constant, that we're assuming very slow accrual at the start of the trial. So after 6 months, we have 2.5 individuals in the trial. And later on, then our trial ramps up, that everyone else is accrued in this period of after 6 months to 18.5 months. We're not using a piecewise hazard ratio, we're just using a constant one. And we see here we get a lesser power from our original sample size. But when we perform a group sequential design like this, we get an output of the big table with all our different details about it. So we can see our outputs. How many events will it be until we're going to perform our different looks. We can see the boundaries for our test statistic, where we're going to stop for futility or efficacy. Here we can see the probabilities for our exits at different stages. So we can see this here, this would be our alpha spending, and then this would be our beta spending. And we can see that clearly here when we come into our error spending plot. We can see a lot of the beta error is spent at the start of the trial. So at the very start of the trial, we're using a lot of this to test for futility. And then we can see our alpha error is being spent throughout the trial and considerably in the later stages, compared to obviously the starting stages, quite low. So in the early stages, we're doing a lot more, we're putting a lot of our emphasis on futility stopping. And we can see that in our boundary crossing probabilities. So we can see quite likely in the case here. And then later on, our efficacy or our stopping for efficacy comes up a bit. And then we can see some details about how our trial would look, depending on if we're under the assumptions of the null hypothesis and the alternative hypothesis. So we can see our trial would probably come to completion a bit quicker under the null, and that makes sense. With a higher hazard ratio, you're going to see more events. If your treatment isn't working as you expected, more events are occurring, and therefore your trial is going to end just a bit quicker. And we can see that our expected events under each of the times under each of the hypotheses. And we can see here just like our expected end times then if we don't cross boundaries in the interim analysis, and then just a summary of our information. The other one I've included here then, I'll just show this off. No differences really, only big thing here now is we're accounting for our dropout. And I suppose it's a good way of showing that this uncertainty around your dropout, that we lose over 10% of power, over 10% power here just on dropouts. And this would be, so it's complicated to me. So this is our treatment group. And we can see, so there'll be a high dropout, the initial in our treatment group, and then we can see a later trend of dropout in our control group. So given those details then, we see a notable drop in power, and we can look at our thing again. We can see, and again, yeah, we see our drop in power considering we're going to have less events due to that dropout at the end of the trial and also throughout the trial, that our interim analysis, we just have less events we just have less of the information to be working with overall. And again, we see our similar information, our boundary crossings and our assumptions in the trial of what would be happening under the different hypotheses. We can see here just that we have our 50 people in the pipeline that haven't experienced an event yet, expectedly at the end of trial and whatnot. So that's it when it comes to group sequential design. We can see how we might perform this in nQuery, and we can see a lot of the different results that can come out. We have some boundary parameters and our design parameters, our expected sample size, which in this case we're going to be expecting the maximum sample size in both our cases. So I'll just go back to the webinar now. Perfect. Okay, so a newly added feature to some of the group sequential design tables is the ability to simulate study data in order to predict group sequential design characteristics. So simulation is a highly flexible tool which allows you to quickly access a given adaptive design over a wide range of scenarios and designs. Compared to analytic derivations, it's a lot easier to access the operating characteristics of a given adaptive design and explore unusual scenarios which may not be covered by the specified hypothesis used for power calculations. Simulation for adaptive designs, such as group sequential designs, has two main benefits. So they're verification and exploration. So verification means simulation can be used to assess if the design actually has the operating characteristics used to generate the initial design. So for example, you can verify your type 1 error, and you can verify your power, the common objectives. Then exploration means using the simulation to quickly assess the operating characteristics under a wide variety of additional scenarios, say for effect size smaller than initially powered, or for generating data from a different statistical distribution. Simulation provides information for both our overall trial as well as the likely outcome at any given interim analysis. And this can be used to make decisions about whether to change the trial. So you might do it for changing group sequential boundary method. So listed here are some of the considerations when it comes to GSD simulations. So step one here is to generate model assumptions, and we have some screenshots here from nQuery on what inputs you may have. So this is for the two-mean GSD. You can see there your design parameters and allotment parameters. So proportion of subjects in each group and how they're randomized, stuff like that. Step two, your design assumptions. So this is the screenshot below. This is sharing some of the values you consider here, like if you want to use pooled or unpooled standard deviation, and if the standard deviation is known, and other stuff like number of looks when it's a two-sided test, and how you're parameterizing your bounds. Your third step will be going to your boundary and information assumptions, and then finally you need to decide just usual simulation variables like a seed number and how many simulations you want to run. This is an example of a group sequential design that we utilize simulation for. I'm not going to go through it today, as Brian does a good job of going through it in depth in the original webinar. So if you're interested in seeing this done out, I would check that out. And again, this is just some more details that we would use in that example. But again, like I said, I wanted to include this just to show that this is something we could do. But again, just for time constraints, I'll just pass that off to Brian's webinars. Again, he does a very good job at going through this. Perfect. So now we're going to move away from all this discussion on survival analysis and group sequential designs, and we're going to take a look at something a bit different, which is our final webinar for today. This is a discussion on non-parametric tests delivered by Dennis in April. So statistical methods have the assumptions for their interpretation to be statistically valid. So you'll often see normality related assumptions for a lot of different tests, or as we've been talking about with the survival analysis designs, you require proportional hazards for some methods. So when these assumptions aren't met, your model can give incorrect or inefficient inference. So you can think, when you think like in terms of requiring your normality assumptions, your t-tests and your z-tests, and if you're not meeting that normality assumption, you're going to be getting ineffective results. So non-parametric tests make little to no assumption about the distribution of the underlying data. So these tests are easier to justify using, as in most cases your data will be compatible, but this comes at the downside then of these methods typically being less powerful compared to, say, you could use their parametric alternatives when you can meet their assumptions. So you'll typically see a pairing, like you'll see an analogous parametric method for your non-parametric method. So usually in most cases, if you can meet the assumptions of your parametric test, you're going to prefer using that over these. So you're better off using them. It's worth noting that some parametric tests are quite robust to distributional deviation. So I know I mentioned there the t-test normality, but the t-test is often a reasonable choice even for quite non-normal data. So you should carefully consider whether you want to use a non-parametric, because in some cases you might still get away with using your parametric example. But again, there's research to be done on that, whether how you want to go about depending on the data you have. So examples where you might use non-parametric methods, you're usually in the presence of complex continuous data. So when you've stuff like extreme outliers or funny variance or whatnot, you'd also use it for ordinal data such as rankings or Likert scales. You'd see them used in that case. So you can see here, for a lot of the common tests, there are non-parametric equivalents. So here you can see your one sample, a pair of t-tests have the Wilcoxon signed rank test. Two sample has the Mann-Whitney U-test. ANOVA has Kruskal-Wallis when you're dealing with factorial type designs, and the Friedman for repeated measures type designs. And then for Pearson's correlation, you have the Spearman correlation as an analogous useful test. So these are non-parametric equivalent or non-parametric tests. These are rank tests, these most commonly used ones. As per their name, these use the relative ranks of the values for each subject rather than using the values from the data directly. So how it usually goes is subjects are ranked relative to all the data, and then rank sums will be calculated, and these will be what the tests will be performed on. And these hypotheses are usually framed around probabilities of one group being larger than the other, or say in the Kruskal-Wallis example, you'll have like a pairwise probability matrix. Rank tests are often conflated with tests of medians, but that's really only valid when you have location shift, which assumes that all your data is distributed in the same way, and the only difference between the groups then is determined by a shift parameter. So if you imagine your distribution shape, all your different groups would be that shape, except you'd slide the distribution shape over the effect size line. Non-parametric methods for testing quantiles, such as medians, are available, but these are not the same thing as rank tests outside of the shift location or strict location shift assumption. Perfect. So when determining sample size for non-parametric tests, methods will typically differ between continuous and ordinal power analysis. This is due to how the methods are structured when dealing with stuff like variance. So for continuous data, you might need to specify a specific data distribution, like when you use the location shift hypothesis. If you can make this assumption, you can gain the power. Like the more you can confidently assume about your data, the better it's going to be for your power, really, and your model. For ordinal data, you may need to specify the proportion of subjects in each rank across each group. From there, you can calculate stuff like probabilities of ranks being higher per group and perform the calculations from these. For both types of data, you can usually either calculate moments from the data. So you might have your pilot data, and from there you'll calculate estimates for your moments. Or you might just have estimates already, either that be from other sources of data or existing research. So these moments, like your probabilities of group 1 being greater than group 2, and etc. Power can be difficult to analytically calculate with these non-parametric tests. So there's a lot of discussion on using simulation-based power to calculate an accurate sample size. If you know how your data is distributed, whether that be from reviewing pilot data or previous studies, you can simulate data and then perform simulation-based power calculations. So this comes with the added benefit then of being able to create confidence intervals for your data or for your results. So we're going to look at two quick examples here that are fairly straightforward. One being for continuous data and the other being for ordinal data. So in this first example here, sample size was estimated using a Wilcoxon rank sum test, using these details. And for the two sample ordinal case, we're using the same test, except this time we're dealing with ordinal data. So our approach is going to change slightly. So we're going to move over to nQuery now to demonstrate both examples. So I'll just, oh, silly me, I didn't get my mouse stuck under a page. Perfect. So we'll swap over to nQuery now. I'm just going to move this over here because I like to keep the results up. Perfect. So this is our first table we're going to be looking at. This is the Wilcoxon rank sum test for continuous outcome. So we can type in our details. Let me just pull it up here quickly now. So we said we're looking for 70 per group was determined using this test. So if we're trying to recreate that, they were performing a two-sided 0.5% significance test. And so as you can see here, we could put in our moment directly if we had access to it, or we can also come down here to this side table and input information that we have available, and it'll calculate similar to how we did it in the survival analysis. It'll calculate and we can put that into our table then. So in our case, we said that the mean time for healing in the control group is 84, and in the treatment group it's 63, and then we have a common standard deviation of 42. And then we can just hit our compute button, and it'll give us our effect size and our probability of x being greater than y. So here we have a 36.2% chance. We transfer that up, and we'll say again our 80% test. And we get 69. And when I said so, we were aiming for 70, we got 69. So whether that be due to rounding or something in the software, we're getting our number that we're essentially looking for. Dennis notes something interesting in the webinar of his, that this Wilcoxon rank sum test has relative, if you have a large sample size, your Wilcoxon rank sum test is going to, of course, you could probably end up using the t-test for large enough sample size, but it has a relative efficiency of about 95%. So if we open up a t-test, I know I'm going to struggle now in finding this. Here we go, t-test for 2. Need test for 2. Yeah, here we go. Okay, wasn't too difficult, fairly straightforward. And we just input the same details, so 0.05. And we put in 4.63. Common standard deviation 42. And our power being empty. We see 64. And if we do 64, if I just pull up distance, the Windows calculator. If we do 64 divided by 69, we get 92%. With larger sample sizes, we're going to tend to this value. The exact value, which Dennis refers to in the webinar, is, I think it's 3/pi. So that comes out at about, I don't know, I don't use this often enough to be able to pull out pi, but I think it comes out to about 95, just over 95%. So it's an interesting thing to note. But as you can see there, that's how we would perform this test anyway for this calculation. In our second example then, we said we're now dealing with ordinal data, so we can see the slight change here. So again, we're dealing with 0.05, two-sided test. And this time we've said that we've got 4, I was going to say 5, but 4. We've got 4 categories, so we enter number of categories. And it's similar to the last table we're calculating a moment, but this time we're going to be using proportions in the groups. So if I was to enter our stuff, and I'm just taking this from the slide, so if you want to recreate it, you can do it using that. But it's nicely laid out in the table. If I could again type this out correctly, but it's nicely laid out that it's fairly easy to put into nQuery from the notes. So there we are, sum of 1. We do the same here. 425 and 0.5 and 0.05. And when we hit our compute, we get our moment here of 54.7%. And we transfer that up and we type in this case, we are going to actually solve for our power. So we're looking for 470 patients per group gets us our power 80. In that case, we see our calculation, our power value got here in that case. So that's just how you would use some of these non-parametric tests. I'm going to bring us back now to our slideshow. Yeah, so that about wraps it up for our discussion on these 3 webinars. Just to recap what we've gone over, we've looked at survival analysis as a tool for time to event trials and how they can be useful in fields like oncology. We looked at some decisions that need to be made when planning a survival analysis trial and how to handle assumptions like non-proportional hazards not being met. We saw how the group sequential design framework can be applied to survival analysis and the complications that it can bring, along with how to use simulation with GSD to verify your assumptions and to test alternative scenarios. But again, I would check out Brian's webinar if you're more interested in that. As again, it's a very thorough go through of that example. Lastly then, we looked at non-parametric tests, what they are, why would you use them, and just some of the difficulties that come in using them. And we saw how to calculate sample size for these tests and the difference when it comes to whether you're using continuous or ordinal data. So that's it for today's webinar and for 2024 really. I hope you look forward to some of the webinars that'll be coming out next year. I certainly do. And as I said, if you're interested in learning more about any of these methods, check out the videos available on the nQuery website. And if anyone's interested in a demo of the software or wants to play around with some of the files featured in the video, feel free to sign up for a free demo of the software. So thanks everyone for attending today's webinar. I'll respond to any questions by email if that's all right after the webinar. And if you have any other questions, feel free to contact us at this email address or visit the nQuery website for more resources. As I said, the slide deck will become available to everyone a few days after the webinar. And other than that, thanks very much for attending and for giving me your time. And I hope you enjoy the rest of your day. So thank you.